With that, I would like to invite Luisa Lanfranco to share her screen. We will stay at the root area. We will look into mycorrhiza. Luisa is an associate professor also at the University of Turin, but at a different department. She works at the Department of Life Sciences and Systems Biology. And her research focuses on plant molecular biology and mycorrhizal interactions. She's also involved in TOMRES work package 2. Luisa, the microphone is yours.
Thank you. Can you see the presentation?
Yes, we can see it.
Okay. Thank you very much, Anna, for this introduction. And today I'm going to present you the result we obtained within the TOMRES project concerning the interaction between arbuscular mycorrhizal fungi and tomato wild relatives. And I move to the next slide. Maybe. Yes. And so we know that fungal communities living in association with plants with different lifestyles are very abundant. Among what we call the plant microbiota, mycorrhizal fungi are probably surely one of the fungal groups which has been studied since a very long time. But under the word mycorrhiza, we actually place different types of symbiotic interactions, which has been originated over a long period of coevolution between plants and fungi. And this coevolution has originated 4 main types of mycorrhizas, and which features specific morphological traits as well as specific combination of partners. We recognize ectomycorrhizas, ericoid, orchid, and arbuscular mycorrhizas. And just to give you some numbers, arbuscular mycorrhizas are the dominant type, mycorrhizal type, with up to 71% of land plants being able to form symbiotic association with a small group of fungi belonging to the glomeromycotina, which are obligate biotrophs. And I just briefly introduce you the process of root colonization by fungi. In the pre-symbiotic phase, the fungus and the plants communicate through diffusible molecules, and so far the best known molecules released by the plant roots are strigolactones. Strigolactones are able to induce spore germination, hyphal branching, and activate mitochondrial fungal metabolism, and also the release of chito-oligomers, which are in turn perceived by the plant and are able to activate plant responses, which are important for fungal accommodation. Then fungal hyphae on the surface of the root can develop these hyphopodia through which they enter the plant tissues, and in the inner layer of the cortex they form highly branched intracellular structures called arbuscule, which are considered the heart of the symbiosis, since they are considered the main sites of nutrient exchanges. In these sites, the fungus release to the plant, provide to the plants mineral nutrients, phosphorus, nitrogen, sulfur, but also zinc, and in turn receive carbo compounds, in particular monosaccharides and fatty acid. It has been recently shown that the fungi relies on the host plant for the acquisition of these fatty acid compounds. So the extent, the level of root colonization, it seems to be under the control of several factors. Some of them are still largely unknown. There could be some genetic, but also some environmental factors. So once established, the symbiosis could provide several benefits to the host plant. And although the association occurs at the root level, we could observe an impact also in distal part of the plants, such as leaves and fruits. We can observe an impact on plant developmental processes, such as, for example, flowering time, and also a higher tolerance to biotic and abiotic stresses. The tomato is a good model to study the IM symbiosis because it responds quite well to the colonization, also at local but also at systemic level. And several studies, some of them also produced inside our lab, already demonstrate the positive impact of the IM symbiosis in alleviating the stress induced by drought. However, the plant responses induced by the M symbiosis are highly variable and can depend on the plant genotypes. In addition, the Solanum genus is also a good model to study domestication processes and to explore the genetic variation within wild relatives. Solanum wild relatives in particular are well adapted to very harsh and desert environment of the ants, as we have seen from the pictures from the Dani presentation. And so in some cases, often these wild relatives display desirable traits which can be useful for the genetic improvement of cultivated tomato. So within TOMRES, our first aim was to analyze the AM responsiveness and susceptibility of a panel of tomato genotypes as well as some wild species. And in particular, we considered also two different growth condition, a control condition and a combined stress, where we induced a combined low nutrients and moderate drought stress. We considered in particular 6 tomato cultivars and 2 wild species, Solanum neorici and Solanum pennellii, and we inoculated them with the well-known symbionts of AM fungi, Phenelliformis mossae. And after a few weeks, we analyzed the vegetative growth, and in particular, we analyzed the growth response and the AM colonization. The mycorrhizal growth response was rather variable across the genotypes and species, and in some cases, we could observe the plant responses were also dependent on the growth condition. Moving to the AM colonization, we could found a rather high colonization level from 60 to 90%. This colonization level in some cases was slightly reduced in the presence of the combined stress. But the most interesting results was that obtained in the wild species Solanum pennellii, this one, because it shows a very, very low level of root colonization. And this was, in fact, unexpectedly. And this low colonization level was associated with a slightly low negative mycorrhizal growth response. We wanted to investigate a little bit more in detail this mycorrhization of Solanum pennellii, and we analyzed the 4 parameters of mycorrhization in comparison to the tomato, the M82 genotype, which was selected as a reference. And as you can see here, the frequency, the intensity, and the arbuscular abundance in the whole roots were lower in the Solanum pennellii compared to the M82. In the control condition, but also in the similar value were registered under the combined stress condition. And this was obtained notwithstanding the number of hypopodia, so the structure that the fungus is using to enter the roots were similar in both species and under the both growth condition. From these light microscope images, you can really appreciate the very strange, very patchy patterns of colonization in Solanum pennellii mycorrhizal roots. And however, within these colonized root fragments, we could observe that the morphology of arbuscules are really similar to those arbuscules formed in tomato mycorrhizal roots. Also, the expression of the AM-inducible phosphate transporters is induced in the mycorrhizal, Solanum pennellii mycorrhizal roots, and this suggests that we are, in this case, we do have a functional symbiosis, since this gene is considered a plant marker for functional symbiosis. We had the chance to analyze the F1 hybrid between lycopersicum and Solanum pennellii, thanks to the collaboration with Dani, and we analyzed the 4 mycorrhization parameters of this hybrid. As you can see here, we register for the frequency, the intensity, and the arbuscular abundance and the whole roots intermediate values for the F1 hybrids compared to the values we obtained with the parental lines. And also the number of arbuscules in the selected colonized roots were higher in the F1 hybrid compared to the two parental lines. So from these data, it seems that the backcross inbreed lines could be a promising tool to identify genetic determinants associated to the extent, to the level of AM root colonization. And to go further and to understand why the low susceptibility of Solanum pennellii to the M symbiosis, we performed also genome-wide transcriptomic analysis by considering the two species, of course, the mycorrhizal and non-mycorrhizal condition, and also the combined stress and control condition. As you can see here from these Venn diagrams, which are comparing the mycorrhizal versus a non-mycorrhizal condition, we could observe a very that Solanum pennellii was very low responsive to the AM symbiosis under the control growth condition, because we could detect only a low number of differentially expressed genes. By contrast, the same pennellii was highly responsive to the M symbiosis under the combined stress condition. So this tells us that this plant species is the response of these plant species to the M symbiosis strongly depend on the growth condition, and this is much more evident than with tomato. We then analyzed and we identified the ortholog gene pairs. We could identify more than 2,000 ortholog genes. And the analysis of the orthologs show a differential pattern of gene expressions. In some cases, we could observe a different and opposite direction of regulation, comparing tomato and Solanum pennellii. And you can see here, for example, these genes in tomato are responsive in terms of upregulation in Solanum in tomato under combined stress, but they are downregulated in Solanum pennellii. We focused on some gene modules which we thought they were important for the establishment of the M symbiosis. And in particular, we found out that the genes involved in the M symbiosis, in particular in the metabolism of phosphate and the lipids, were actually expressed and equally responsive in the two species, as you can see here. But with some exception, because some genes related to the symbiotic signaling pathway were not responsive to the M symbiosis in pennellii compared to the tomato. In addition, also, tomato and pennellii show a differential regulation of genes related to the strigolactone metabolism. For example, you can see here how these genes related to the biosynthesis of strigolactones are responsive to the M symbiosis in tomato, but not responsive in pennellii. And under combined stress, we have an opposite regulation with a down regulation here and up regulation in pennellii. So these data suggest us that maybe the low susceptibility, the low colonization level in Solanum pennellii could be due to a low production of strigolactones, because we know that the crucial role that strigolactone plays in the establishment of the M symbiosis. So to verify this hypothesis, we perform an experiment where we treat Solanum pennellii mycorrhizal plants with GR24, which you know is a well-known strigolactone synthetic analogs. And we analyze the frequency, the five mycorrhization parameters. There is a trend, but only in one case we have an increase in one of these parameters. For the rest, we can say that this GR24 treatment does not actually increase the mycorrhization in Solanum pennellii. We set up another experiment, which is called the donor plant experiment, which is a, let's say, common practice in mycorrhizal research to understand whether mycorrhizal plants surrounding another plants could provide stimulating compound to increase mycorrhization. So in this case, we set up some microcosm where the Solanum pennellii plants were put here in the middle and were surrounded by other mycorrhizal plants, in this case other Solanum pennellii plants, or in this case we use 5 mycorrhizal plants over a rice line which is known to overproduce strigolactone. And then after some weeks, we again measure the 4 mycorrhization parameters in the central pennellii plant. And as you can see here, for at least for 3 out of 4 mycorrhization parameters, we could see an increase of the percentage. So the mycorrhization level in pennellii could be increased by surrounding the plants with a strigolactone overproducing rice line. Of course, we were curious to see whether the tomato and pennellii produced a different amount of strigolactone. For this purpose, we used a biological assay based on the Orobanche egyptiaceae seed germination test. As you know, the seeds of these parasitic plants can germinate upon the perception of strigolactone. And this biological assay is widely used to detect and also to quantify the amount of strigolactone. So we collected the root exudate from the 2 species that were grown under hydroponic condition, and we used the root exudate to treat our Orobanche seeds. And then we measure after a few days the root germination, the seed germination. As you can see here, we could have a 60% germination, seed germination for the GR24, which was used as a positive control. But we could observe about 30% germination rates by using root exudates from both species. And we have here 2 different samples for each species because we collected root exudate at 2 different time points. So at least based on this biological assay, we can exclude that Solanum pennellii is not able to produce or not enough strigolactone, because it seems that the exudates of pennellii show parasitic seed germination activity similar to those of tomato exudates. So in conclusion, our data demonstrate that the responsiveness to the IMB symbiosis can differ across accession and less often under our combined water nutrient stress. And this responsiveness to the symbiosis is not strictly correlated to the colonization level, which is something that confirms some data already present in the literature. And Solanum pennellii turned out to be a very interesting case study because it shows an unexpected and very reduced and patchy AM colonization. However, arbuscular morphology and possibly the functioning of the symbiosis in these very low, little colonized root pieces is normal. Concerning the transcriptomic data, we clearly show that the 2 species show a very different modulation of gene expression upon AM colonization. And Solanum pennellii, in Solanum pennellii, we could detect very few mycorrhizal responsive genes under control condition, while many mycorrhizal responsive genes, more than 4,000, were detected under the combined stress. Notwithstanding, we had similar colonization levels in the 2 conditions. And we could observe a differential regulation of strigolactone-related genes. But despite the fact that we can increase the mycorrhization in Solanum pennellii by surrounding it with the strigolactone overproducing plants, at the moment we can exclude that Solanum pennellii is not able to produce enough strigolactone in the root exudates. And so this is still remain an open question. We do have some ongoing work. We would like to deeper analyze the strigolactone composition in Solanum pennellii, root as well as root exudate and extracts. And we also would like to evaluate the susceptibility of pennellii to other arbuscular mycorrhizal fungi, and also to explore what occurs on the fungal side by looking at the RNA-seq data. And of course, we are willing to analyze the backcross inbreed lines, possibly to identify genes QTL associated with the level, the extent of mycorrhizal colonization. And with this, I would like to acknowledge, to thank the people who works on this project, especially Matteo Chialva, who did the majority of the work, all the people in staff at the Unito di Bios, and of course all the students that also participate by working on their master thesis. And of course, I would like to thank also the TOMRES Consortium for the precious advices and the material, of course, Dani Zamir for providing us the seeds. And I thank you for your attention.
Thank you very, very much, Luisa, for this very interesting presentation, going deep down into the roots and the genetics of the mycorrhizal colonization. Thank you very, very much.
You're welcome.